All right. Here goes. This is going to be the last lecture. We're done, basically. There's just a few more items to go over, and then you can use the rest of the time to study for all your exams coming up. So, we are basically, all we have left to finish is sort of this types of inheritance stuff. We looked at dominant recessive already a little bit. You saw this table, the dominant disorders versus recessive disorders. Some of the dominant ones can be pretty rare if it's a disorder like Tay-Sachs or Huntington's. Huntington's, achondroplasia, syndactyly, and so on. Because dominant things can be relatively easily eliminated from the gene pool, because if you just have that disorder allele, then you express that in your phenotype and you may not do so well. The ones on the right side, the recessive ones, are a little bit more insidious because people can be carriers of recessive alleles and they're perfectly healthy, but they carry that recessive allele to pass on to their children, maybe in some cases, and so the recessive ones are not as easily eliminated by natural selection. And we looked at example of doing these Punnett squares and being able to figure out probabilities of offspring based on this dominant recessive inheritance. We also have something called incomplete dominance, and this is the first new thing here. Incomplete dominance. This is when you get intermediate phenotypes in the heterozygotes. So you have two versions of an allele, and somebody has a copy of both versions, and the phenotype is kind of intermediate. Like a good example of that in plants would be flower color. If you have a plant that has red flowers, and another one that has white flowers, and you cross-pollinate them, and the new plant is pink. Okay, so that would be a good example of incomplete dominance of flower color or flower pigmentation. In humans, a good example of this is sickle cell. I tried to explain this before with this intermediate phenotype. So somebody who has two copies of the sickle cell globin gene is going to have a real hard time and bad outcomes because they get sickle cell episodes when they're stressed for oxygen. So if they go to elevation or even starting to work out, can bring on a sickle episode and their outcome is not that good. And if you have two normal globin copies, then you're okay. But if you go into an environment where there's lots of malaria, you're actually not doing as well as people with the intermediate phenotype having one of the sickle globins, one of the normal, that makes their red blood cells resistant to the malaria parasites which live inside the red blood cells. So there's an intermediate phenotype, and really if you were to look at their hemoglobin, half of it would have that sickle globin, half would have normal globin. So they're really in between, and nothing is completely covered up. So that's the only example I have here from people for intermediate or incomplete dominance. Another one is multiple allele inheritance, and that's anything if you have more than two versions, more than two alleles, and so three would be a good example. And the example I have is the blood types. The A, B, O blood types are actually based on three alleles. And any one person has two of those three alleles, so you can put them in various combinations to come up with our blood types, and this is how the alleles are usually written. So you have capital IA, capital IB, and lowercase I. So for A, B type, and then the lowercase I would be type O. And if you're type A, if you have type A blood, you can be one of two genotypes. You can be either both for the type A blood, or one allele for the type A blood, and then one of the recessive alleles for, basically that's type O. You have nothing on the surface of your cells, if you remember how that works, right, with the typing. Type A cells have these A guys on their cell surface. So there's a gene for that, and that's this one. So either one can be for type A. And then here's a table that has all the combinations, table 29.2. So type O blood is these two. Type A blood, you got these two options. Type B blood, those two options. And then AB, you have one of each of the dominant blood typing alleles. And if you're a type AB, you know you have to get one of these from your mom, one of these from your dad. And so if you know your blood types in the family, you can see if all that works out, if you know these. Okay? So if the A came from your mom, well, your mom could have been either this or that, right? To donate an A. If your dad gave you this one, he could have been either this or that. And if you're type A, okay, you don't know what you are, but then maybe if your mom is type O, then you know your mom would have been giving you that one. That's all she had. So you're this genotype, and then you know your dad had to have had that A in there somewhere. So you can do pretty good scenarios like this. We've actually had in Dr. Super's genetic class, I don't know if I mentioned this before, twice now in I don't know how long she's been teaching, maybe 12, 15 years, somewhere there, where people found out that their dad wasn't really their dad. From paying too good attention in genetics class. So you do your blood typing, and then you learn about this, and then like 3 days later they come back to her, it's like, I think there's something going on. So you can do draw all kinds of genetic conclusions from this.
So if I know that I'm A and my mom was O, then I would know that my dad was A.
Yeah. Yeah. You know, and there's some impossible combination, right?
Yeah.
Sex-linked inheritance. This is when you have genes, usually it's the X chromosome that we're talking about here. Genes only found on one of the sex chromosomes. Genes only found on the X chromosome usually, and in sex-linked cases, when something is sex-linked on the X chromosome, it tends to be much, much more common in men than in women. Women can have it also, but it's going to be very rare. And if something is on the Y chromosome, then you already know, well, that cannot occur in women. So let me explain this a little bit. For example, a lot of the hemophilia cases are sex-linked, where genes for clotting factors are located on the X chromosome. And if you have some sort of mutation in a clotting factor gene on that X chromosome, if you are a man, since that's your only X chromosome, you don't have another one to make up for it, you're going to express hemophilia. A woman may have a normal version of that clotting factor on her other X chromosome to make up for that. Okay, so any sex-linked inheritance thing on an X chromosome is going to be much more common in men than in women, and that is the case for hemophilia. It's called sex-linked because it's the sex chromosomes. The last one is polygenic inheritance or polygene inheritance. Polygenic inheritance. This is when a phenotype is dependent on more than just one gene, like a lot of them are. One really nice clear-cut example is skin color. So how much melanin is your skin retaining, and that is a nice example of polygene inheritance. And here's a graph for that, figure 29.6. We have 3 genes for skin color. They all basically do the same thing. It's melanin production, involved in melanin production. And here we have some hypothetical genetic combinations. So we have a set of parents, and the A's, B's, and C's are the skin color genes. And if you have the uppercase, that means you're making a lot of the melanin to deposit. And if you have the lower version, your melanin output is very minimal. So here we have the extreme version for very, very light-skinned, where all the alleles are of the low melanin output variety. So you have all lowercase letters, 3 genes, A, B, and C. And then you have AA means the 2 alleles on the chromosome pair, right? So you have 3 pairs of genes. And this individual has all the dominant ones, so it's very, very dark. If you take the first generation offspring, they'll all be completely intermediate. They have to be, right? Genetically, you can do Punnett square for these 3, A, B, and C. And this parent can only give lowercase, this parent can only give uppercase versions for all of them. So they're all going to be this intermediate genotype, one of each, and they'll be intermediate in skin color. What's interesting then, when you take that to subsequent generations, obviously you wouldn't mate sisters, brothers, but another offspring from a similar pair of parents, you could get this kind of variation in skin tones. If you had 64 offspring, which is obviously not possible, but if you had, those would be the probabilities. So you had 1 chance of 64 to go back to very light grandparent. You had 1 chance of going back to the very dark grandparent, and then you see the probabilities of the different range of skin colors. So you can get a very large range of skin colors with the 3 genes and those allelic combinations. You can actually make a Punnett square that has 64 squares. If you take genetics, you probably have to do this at least once, one of the big ones. Anyone did this in genetics? Big giant one with the 64. And what this is accounting for is when you think about African Americans, these are all sisters, okay, from the same parents. And what's kind of funny, these people had 2 sets of twins in a row. That's very rare, but there is a link. When somebody has a set of twins, they're more likely to have more. And they're obviously fraternal twins, not identical, or the skin color would be identical, obviously. But these 2 girls, twins, look how different their skin color is from that just random combining of these skin color genes. And their younger siblings, same thing. So you get this from this combination of this sort of dosage of the polygene inheritance. You get this very commonly. And in African Americans, obviously from slavery times onwards, with first the forced intermixing and then ever since just the random mating in the population, it's very common to have very differently colored babies. to the parents from this polygene inheritance. So that was this one, and then that's it for inheritance types. And a couple more things. So there's another factor that can have an influence on hereditary, and that is what we call phenotypic plasticity. And this is actually kind of a big deal, not a small impact, because the environment actually does shape or influence the expression of our genes. And it's always a nice example when you think of plants. Again, you can have genetically identical seeds and you can grow them up into plants in very different environments, space, different water, different nutrients, different sunlight, and the plants will look very, very different once you're done growing them up. And similar things happen even in people. When you look at the environment that we have inside the uterus that a baby is exposed to, and then even after birth. So we have, as an example, toxins that can negatively impact development inside the uterus. These toxins are called teratogens. Teratogens causing birth defects. So teratogens cause birth defects. So there are some drugs you're not able to take during pregnancy because there are birth defects associated with it. For example, high amounts of retinol. There's some acne products that you have to sign a sheet that you're not going to get pregnant, that you're going to take birth control of a certain kind, to get that, the Accutane, or Accutane, I don't know how to pronounce it. Because it has severe birth defects, possibility if you take that and you get pregnant. A historical example is thalidomide from the 50s and 60s, when women took it against morning sickness, and then they had babies with no arms, or extremely shortened arms, where the hands a lot of times were attached right to the shoulder, that was a pretty famous, everybody hears about that in genetics, I think, thalidomide. And there are many others. Alcohol is another pretty bad one. Excessive alcohol during pregnancy has a serious impact on brain development in the infant, and even facial features, fetal alcohol syndrome. So lots of different things that can go wrong. And that has nothing to do with your genetic makeup. It's how that is being able to be expressed in the presence of these drugs. Also, malnutrition also has an impact. So even after you're born, malnutrition can really impact your phenotype, no matter what your genotype would be under. And if you're able to grow up under perfect conditions, that's great, but with malnutrition you're not going to get as tall. For example, there are old studies from Mexico where they would supplement some siblings and not others with vitamins. There's the traditional Mexican diet that relies so heavily on corn and beans. There are some minerals and vitamins that are missing in that, and they were supplementing some of the siblings but not others. And this goes back into the 50s. Nowadays, nobody would do stuff like that. It's not ethical, but there's a lot of stuff that was done in history that we wouldn't do anymore. But we still learned there that malnutrition really had a big effect on height. And you can also look just at populations where malnutrition, the people are shorter on average. And genes also interact, so you can have genes that are affected that are not directly related to the expression of the phenotype, but there's something downstream from them that they do affect. And so genes don't really act in complete isolation, so they do affect each other. All right, with all this genetics, if you have genetic traits running in your family that may be undesirable, like cystic fibrosis might be one, you can do genetic screening and counseling. If you're interested in the likelihood of having a child that might be affected with cystic fibrosis. And genetic screening and counseling. So one of the things you can do is carrier recognition and pedigree analysis. And for some traits, you can learn a lot from that if you know in your family history what's going on there. So you kind of have to know what's happening in your family history. Carrier recognition, pedigree analysis. This is a pedigree that genetic counselors will be able to draw if you have a lot of information on your family. It's basically a family tree. The square shapes are the males, the round ones are the females. And I just got this off the internet. So this is about sickle cell trait. So here is the black color denotes sickle cell gene, sickle cell trait. So if you're fully colored in, that's bad news. You have the sickle cell disease. But if you're half and half, you're going to be mostly okay, but you're still a carrier. And so you may wonder if you're going to pass on this sickle trait to your offspring. What are the chances? And, so you can kind of look at this, okay, I'm not having the trait, I'm good or not, depending on your family. And you could do this for a lot of different things. Let me see, what's the next one? This is something about neurofibromatosis. I don't even know what that is. But another pedigree. So they're wondering about Melissa, if she's going to have this trait, and they're going back in the family. On this side, nobody here had this. It's only from one side of the family. And so they found out, well, okay, her dad didn't have it, so she's good to go. So that's the kind of thing people get during genetic counseling. And then some things can also be found through fetal testing, and more. Every year there are actually more genes that are actually discovered for various diseases. So people are able to pinpoint what the gene is that causes stuff, and then you can do fetal testing. The pictures here are all for fetal testing after you're already pregnant. What's not illustrated here is any testing you can do during in vitro. I mentioned that how you can suck out cells from that inner cell mass, and it's not going to be a bad effect. So this is after you're already pregnant. Maybe you're above average age, you're really worried about stuff. You can do this. Amniocentesis is pretty invasive, and it can trigger labor and introduce infection. Here we have to be relatively far along, 14th to 16th week, because you got to have enough fluid surrounding the baby that you're poking literally through all those layers, so through the body wall into the uterus, so through the uterine wall into that amniotic sac. So that it's a little bit risky doing this. Then because the fetus is shedding skin cells already, even at this early stage, you can get amniotic fluid, spin it down in the centrifuge to collect the cells, and then grow them up in lab as a cell culture until you have enough to do testing, and, for example, karyotyping to see if there's a chromosomal abnormality, if you have Patau syndrome or anything like that, or Edwards syndrome, where it's an abnormal number of chromosomes. Most of you probably know about Down syndrome. It's an extra chromosome 21, but there are some much worse conditions than Down syndrome with extra chromosomes. And that you can decide what you're gonna, if you're going to continue the pregnancy or not, depending on what kind of thing comes up. Earlier is better generally, and chorionic villus sampling can be done as early as 8 to 10 weeks because you're taking samples of the fetal tissue from the fetal proportion of the placenta, from the chorionic villi, and then you also don't have the delay of having to generate enough cells in cell culture. You have enough cells right away to do the genetic tests like karyotyping on here, or testing for some other genes. So it's earlier and it's faster, and most people prefer this this type of testing. And that's the same thing as with amniocentesis. Then you can decide if there's a doomed pregnancy happening or not, potentially. And a lot of these chromosomal abnormalities are doomed pregnancies. So really, there's no chance for development to continue. So those are the options for fetal testing if you have concerns. And that is all I have, so I am done. But I did bring some practice questions. Do you want to see some practice questions since you're here? Body, butt, and chair. You want to stay a little? So let's do that. And let me talk a little bit about the final it will be just like what we did for AMP1, so I'll have 50 questions on the material since the last test, which in my estimation is digestive, right? Digestive, urinary, reproductive, pregnancy, hereditary. Is that right? Is that what? I'm pretty sure, right? So starting digestive wasn't on the last test. Urinary, reproductive, pregnancy, and hereditary. So, has anybody studied anything yet? And our final, I believe, starts at 2 o'clock. So you got to remember that, and it's kind of on the wrong day. It's on a Thursday. So you got to remember not to show up on the wrong day.
What time is it at? 2?
I think on next week Thursday at 2, in here. And so 50 questions on that new stuff, and then I'll have 25 old random questions from my test bank. So I don't know exactly if you've seen them before or not. Chances are there's some of both. Okay. How did it go with the lab final, actually?
Some of us are very happy, others wasn't so much.
So as soon as I get those numbers from Ely, I will update the grade sheet. Okay, so you can plug in. I assume I'll get them in the next few days. This is not such a big group anymore. So keep an eye out on the grade sheet.
Some of us hang out after class.
Yeah. Okay, do you want to go over some of these?
Yes.
Intestinal villi are found in which of the layers of the alimentary canal? So you'd have to know the layers. The villi are B, yes, in the mucosa. That's the innermost one. What's the next one out from there? Submucosa, and then muscularis externa, and then visceral serosa. That's right. So B. How about the saliva? Which of the following is true about saliva? C. That's right. What does that digest? Carbs. Okay. Good deal. Initial portion of the small intestine, duodenum. That is followed by the jejunum, ileum. All right. So you did do something. This one, hydrochloric acid secreted by
Chief cells.
think it's the parietal cells.
Parietal.
Which region of the nephron is most likely to be found in the cortex? Loop of Henle, collecting duct, renal pelvis. That makes no sense, right? That's not even part of it. D, proximal convoluted tubule. Yeah. What else is up there? The what? The glomerulus? Yep. What else is up there in the cortex? Distal tubule. What's the other part of the kidney called, where the loop of Henle goes into? Medulla. Right. Descending limb of loop of Henle, not permeable to water, freely permeable to sodium. I didn't really talk about urea with you all, so ignore that part. Pulls water by osmosis into the lumen of the tubule, contains fluid that becomes more concentrated as it moves down into the medulla. Yes, D. So first we're sucking water out, and then we'll suck more salt out. So it gets first more concentrated and then it gets more dilute, but far less of it. So D. 60% of the synthesis of semen. Are there some nonsense ones on there? Yeah. Which one's the nonsense? D. Yeah, all of them?
All of them but A.
Yeah. A. Seminal vesicles, or I think now they call them the seminal glands. The female zygote received chromosomes from the mother only, the father only, both mom and dad, and none of the above. That's kind of a...
It's a gimme question.
You'd be surprised. I've sometimes had to give the answer to questions, and still there would be one person not having it marked. C, right? Yes? That's a trick question. This is another really tough one. A normal female is XX, yes. All right, when we ingest, I don't know why it goes back to digestive, but kind of random. When we ingest large molecules such as lipids, carbs, and proteins, they must undergo reactions whereby enzymes split these molecules. This series of reactions is called chemical digestion. Yes. Where do most of these enzymes come from? Just in the bulk, lots of the digestive enzymes, where are they coming from? What makes them? Are they just sort of in the gut? How do they get to the gut? Digestive enzymes are made by... Somebody's got to have something. How about the pancreas? Ever heard of that thing?
Yeah.
It's like one thing, if you have pancreatic cancer, you can't live without it.
Yeah.
Yeah, it's kind of a big deal. Pancreas. What's the liver have to do with digestion then? Isn't that also involved there somewhere?
Yeah.
Bile, yes. It makes bile. Well, then, digestion of which of the following would be impaired if the liver would stop functioning? B, lipids, right? Bile, lipids, yes. Emulsification. How about the dental formula? 2, 1, 2, 3. What's the 2? This two? No. Incisors, what's the one?
Canine.
Canine, the two? Premolar? Molar, all right. What's the maximum number of teeth you should be getting in your mouth? 32. There's another kidney one. The renal corpuscle is made up of, and there's a name change. It's not, you don't have it in the new book as Bowman's capsule.
Yeah, I was gonna say.
It does say it.
I don't know, they eliminate all the references to people. It's a strange
It's in
thing.
parentheses or something. It's in there.
Oh, do they still have it? Yeah. Renal corpuscle capsule, glomerulus inside. Excretion of dilute urine requires dilute urine, lots of dilute urine, okay? Requires permeability of the tubules to water, impermeability of the collecting duct or tubule to water, transport of sodium chloride out of the descending loop of Henle, presence of ADH, copious dilute urine. How do you get there? Is ADH making you pee a lot or less? Less, right? So that's not it. These are tricky, aren't they? The kidney ones. So it's probably a good idea to go back to what happens at every step. Anybody want to venture? How does ADH act in there? It definitely has something to do with water, right? Yeah. So ADH makes you want to hang on to water, doesn't it? How does it do that? No takers. ADH can attach to aquaporins, channels for water in the collecting duct. And when it does that, then water is allowed to leave the collecting duct, leave the urine. So when you don't need to hang on to water, you close all those water channels and make the collecting tubule impermeable to water. So all that water has to leave. You're not reabsorbing it. So B. What's the female counterpart of the male gland's penis?
C.
Clitoris. Okay. The labia majora have a counterpart in the male. What's that? Not the foreskin, nope. The labia majora have a counterpart in the male too. Scrotum, right. Implantation, when does that occur? 1 day after ovulation? No. 2 days after fertilization? No. 7 days after ovulation? Approximately, okay? Just before the onset of menstruation, not usually. It usually already happened there. All right, during the secretory phase of the menstrual cycle, LH reaches its highest levels, progesterone levels are at their highest, estrogen is at its highest, the vesicular follicle forms. That's a tricky one, isn't it? There'll be several of those hormone questions. All right, I only have one of them on here, I think. Progesterone. look at those graphs and how they match the different. And I have another hormone one, but the next one is easier. Oh, there it is already. But prolactin. Or do you want to go back to this one? Like progesterone, what makes progesterone? Does anybody know? What's the structure called that makes progesterone? Corpus luteum, right. So that shows up only during the secretory phase. LH comes from the pituitary that triggers ovulation. The vesicular follicle is not during the secretory phase, that's before. So that doesn't match. And estrogen gets high during the follicular ripening phase too. So it's not at its highest levels right there. All right, prolactin, important. Which of the following is correct? Drop in prolactin causes onset of menstruation? No. Prolactin triggers vigorous contractions during labor? No. Milk letdown is caused by pulsatile prolactin release? No. Prolactin stimulates breast enlargement and milk production during pregnancy and later, really, too? Yes. 19, easier. At which stage of labor is the afterbirth? C, placental. When is the baby expelled? Expulsion. Exactly. All right. Some genetic ones. Good. One possible explanation for siblings having very different skin color is, we just talked about it, polygenic inheritance, continuous phenotypic variation. Bell curve. Most are going to be in the middle, but you can go to the extremes. Some people may be in the sun longer. Well, the condition of having two alleles expressing two different versions of a trait. Heterozygous D. And and then I just grabbed this out of a file. So I don't know why these are there, but might as well do them. See, the middle portion, yes, jejunum. Let's do the bottom ones. For cart digestions initiated in the mouth, which this goes back to respiratory and blood. So how does carbon dioxide or oxygen interact with pH? Does oxygen have anything to do with pH? No. But how about carb? Is low carbon dioxide low pH, or is it high carbon dioxide low pH? High, yes. See? High carbon dioxide. Low pH, because the CO2 dissolves in water, carbonic acid, low pH. And then the filtrate one, that's a good one for just testing overall anatomy. It's kind of annoying because you have to read them all. Ureter, lubricant, collecting duct. Okay, which is the first one on here? Bowman's capsule. Then proximal tubule. Then loop of Henle, then distal tubule, then collecting duct, ureter, urinary bladder, urethra. Okay, that is all I have. So we don't have to meet till the final. If you have questions, just email, see me. Okay? And good luck in all your other stuff.